Welcome back, everyone, to the second annual Mito2I Research Symposium for the afternoon sessions. We have an exciting afternoon for you discussing mitogenetics and then mitochondrial innovation. So today, to start off the SEC session, is Dr. Vanessa Gonclaves. She was a scientist in the brain and therapeutics at the Center for Addiction and Mental Health. Vanessa received her PhD in biochemistry and immunology from the Federal University of Minas Gerais in Brazil. She then moved to Toronto and completed a postdoctoral fellowship in molecular brain science at CAMH, where she is now a project scientist. She has multidisciplinary background in genetics, molecular biology, statistics, and epidemiology. Her research interests lie in advancing research on the role of mitochondria, gene variants in risk and phenomenology of neuropsychiatric disorders. Please join me in welcoming Dr. Vanessa Gonclaves.
Hello, hi. So I'm Vanessa Gonclaves. I'm independent scientist now, so upgrade. I'm independent scientist at CAMH and also assistant professor in the Department of Psychiatry. Right, so I work with mitochondrial genetics. That is the field where we analyze variants in human mitochondrial genes. That includes genes in the nuclear DNA as well, important genes for bioenergetics that's located in the mitochondrial DNA. Right, so mitochondria, as you know, it's involved in many important jobs. It's the main source of energy for the cells. It's the main source of reactive oxygen species. Controls cause metabolism in the cells, apoptosis. So with all these important jobs, we can imagine that variants in mitochondrial genes can lead for a range of disease, from metabolic diseases to neuropsychiatric disorders, right? So mitochondrial genetics is a good target for all complex disease that involves deficits in energy or neuroinflammation and many others things involving mitochondria, right? So today I will introduce two students, and the first student is Melissa Geng. She is a PhD candidate. She works with Niels Sonheimer's lab in the Department of Pediatrics and Molecular Genetics, the hospital for sick children. The title of her presentation today is the Guanine quadruplex structures in mitochondrial health and disease. So I will pass the word for Melissa.
Hello, thank you for that lovely introduction. Hi, everyone. My name is Melissa, and I'm a PhD candidate in Dr. Niels Sonheimer's lab at SickKids. Today, let me tell you a little bit about my project on exploring the role of guanine quadruplexes in mitochondrial disease. So mitochondrial disorders lack targeted therapies. Supportive care is currently the only treatment option available. Today, I want to discuss something called heteroplasmy shifting as a potential therapeutic strategy. As there are multiple copies of mitoDNA within a cell, it presents a phenomenon called heteroplasmy, which is defined as the coexistence of different mitoDNA alleles within the same cell. Heteroplasmy load has been shown to correlate with disease penetrance and severity, and so could potentially act as a target for treating mitochondrial disorders. For example, if we have a cell with multiple copies of the pathogenic allele depicted here in red, if we can somehow target these pathogenic copies of mitoDNA to prevent their replication, we can then promote the replication of wild-type mitoDNA alleles and hopefully restore mitochondrial function. So mitoDNA is unique from the nuclear DNA in that it has a heavy strand enriched in guanines. These repeated polyguanine tracts allow for the formation of a type of DNA secondary structure known as a guanine quadruplex, or a G4 structure. So looking at the figure on the left here, to help orient you, the outermost tract shows mitoDNA in a counterclockwise fashion with a control region at the very top. The innermost tract in green is a position of stable G-force predicted to form along the mitoDNA sequence. The next tract in yellow shows the predicted unstable G-force structures. So what I want you to take away from this figure is the amount of G-force predicted to form through mitoDNA. G4 structures have been shown to decrease polymerase processivity, and especially of pol gamma, which is a mitochondrial polymerase. So shown here is a polymerase stop assay. We can see that with the addition of both pol-G subunits, we get nice extension of a non-G4 sequence. However, with the G4 sequence, we instead get a lot more of the shorter stopped products. This effect is abolished when the extension is carried out in lithium chloride, which disfavors G4 formation. So on the previous slide, I told you that polyguanine tracks can form secondary structures called G4s. So then what exactly are these G4 structures? G4s are a type of non-canonical DNA secondary structure where four guanines come together in the same plane to form a tetrad through hooksine base pairing, as shown by the gray square on the figure on the right here. The multiples of these tetrads stack together to form a quadruplex structure. These G4 structures have been shown to decrease polymerase processivity, and some pathogenic mitoDNA mutations result in an increased propensity for the sequence to form a G4 structure. As an example, I'm showing you here on the bottom the sequence around region 10191, which is a pathogenic mutation leading to a disease called Lee syndrome. The pink bars denote guanine tracks that can participate in hooksine bonding to form a G4 structure. The position of the T to C mutation at 10191 is marked in red. So with the C mutant, the sequence can actually form two types of G4s. The first form shown here, where any guanine in this long G track, second from the left, can participate in binding as one guanine stretch, or alternatively, it can be considered, sorry, my slides are not, there you go. Or alternatively, it can be considered as two guanine stretches connected by a loop size of 0. Previous work, okay, there we go, sorry. Previous work in the lab has identified 6 pathogenic mitoDNA mutations that result in an increased length of guanine tracks on mitochondrial DNA. So underlined here are the guanines that participate in the formation of G4 structures, while the site of the mutation itself is highlighted in red. So all of the mutations here increase the length of the guanine stretch, which enables the sequence to form a more stable G4 structure compared to the wild-type sequence. So for my project, I'll be focusing on the T to C mutation at position 10191, which results in a non-synonymous serine to proline loss of function mutation in the protein ND3, leading to a disease called Lee syndrome. Previous work in the lab has found that non-specific G4 binding agent berberin is capable of inducing heteroplasmy shift in patient cells. On the x-axis here is time in weeks, and on the y-axis is the change in % pathogenic allele compared to week 0. Over an 8-week treatment period, we can see an upward drift in % pathogenic allele in the control cells, which is what we typically observe with these patient lines. Then with the berberin-treated cells, the same upward drift is not observed, and the % pathogenic heteroplasmy actually decreases compared to week 0. So this really made us wonder if we could identify novel G4 binding agents with high specificity to 10191C, which could then potentially be developed into therapeutic treatments for Lee syndrome. So this is our current pipeline for the identification of selective G4 binding agents. We start with a biochemical screen, which is done in collaboration with our partners at the National Cancer Institute. So for the screen, small molecules are affixed onto a microarray, and a solution containing fluorescently tagged oligos is flown over this microarray to allow binding of the target oligo. Compounds with high binding affinity will retain the oligo and show up as spots with brighter fluorescence when imaged. So on the right, you can see an example output image. We quantify the difference in fluorescence between the inside of the white circles and the outside as a measure of oligo binding. So far, we've screened over 20,000 compounds with the small molecule microarray method. Candidate hits are compared to a panel of controls, including single-stranded DNA, double-stranded DNA, single-stranded RNA, other DNA secondary structures such as a replication junction, as well as other DNA G4 structures to ensure specificity to the 10191 G4. From our most recent screen, we obtained a hit rate of approximately 1.3%, of which 0.4% are selected for the 10191C oligo. We then look at these compounds to remove those that are metabolically active or are difficult to do chemistry on. Next, we would use an in vitro primer extension assay to characterize the candidate compound's ability to inhibit polymerase processivity. We mix the 10191 template with P32 labeled primers, then add intact polymerase to allow for extension. Extension will result in two types of products: the full length, where the polymerase was able to replicate past the G4, and a truncated stop product, where the polymerase is stalled by the G4 structure. So this is an extension from a candidate compound from our previous screen. We call this compound C50, and it can potently inhibit polymerase processivity through the 10191 G4. So going through the plot slowly, the first lane is a negative control with no G4 forming propensities. So the topmost band you see it in this lane represents the full length product. So here we see no stop products, then a faint band for leftover fruit primers, and a strong band just above 20 nucleotides, which represents the loading control. The next lane is a ladder, then a positive control optimized for G4 formation. With a positive control, we see no full-length formation. Instead, we see a band just above 30 nucleotides, which is one of the stop products. The next 6 lanes are the wild-type T sequence in increasing concentrations of C50. We see a gradual loss of the full-length product formation and an increase in the band intensity for the S1 product at higher concentrations. Similarly, with the C oligo extended in increasing concentrations of C50, we see a disappearance of the full-length product and an increase in both stop products. So for compounds like C50 that display potent inhibition of polymerase processivity, we then move to characterize their cellular localization. So this is an example with berberin, where since the compound itself is fluorescent, we were able to use co-localization with MitoTracker as a measure of whether berberin localized mainly to the mitochondria in cellular contexts. Lastly, we would characterize the cellular effects of these compounds. We would look at mitoDNA quantity levels, as well as the change in pathogenic allele heteroplasmy over an 8-week treatment period. We also use seahorse as a measure of mitochondrial function to see if prolonged treatment can improve the disease phenotype we see in patient cells. So in summary, I showed you today that the strand bias in mitoDNA makes it prone to form these secondary structures called Guanine quadruplexes, which are capable of inhibiting polymerase activity. This could then be utilized to induce heteroplasmy shifting of pathogenic mitoDNA alleles, as was observed with the promiscuous G4 binding agent berberin. I've told you a little about our current pipeline for discovering novel G4 binding agents, and I've showed you a little bit of results from a recent small molecule microarray screen. With that, I would like to thank my supervisor, my committee members, as well as my lab members that have contributed to this project, especially Mansur, who did the work related to berberin. I would also like to thank Mito2I for their support of this project, as well as this wonderful opportunity to present my work here today. So thank you all so much for listening.